Are there homologous chromosomes in this picture?
Yeah. Mm-hmm.
Okay. Where?
Except for the autosome, except for possibly the sex chromosome.
Okay, so let's talk about that's the easiest one to pick out, the X chromosome down here in the bottom. That's 2 homologs. Those are homologs. That means it's 2 different copies of the same chromosome, 2 X's, or 2 chromosome 1s or 2 chromosome 2s. Each of our cells were diploid. We have 2 copies of every chromosome. So when I say 2 copies of every chromosome, which I've said a lot in the past few days, what I could say instead is homologous chromosomes, which means that one of them came from your dad, so chromosome 1 inherited paternally, and chromosome 1 inherited maternally. Simple. Now things get kind of weird. How many copies, how many molecules, let me ask first and let people think, and then I'll take your question. How many molecules of DNA have I circled here in blue? So think about that, and bless you, and yes?
Is that supposed to be on the January 30th slides? Or is that
so I just added that on the fly. So you'll see a blank slide without this figure. I'm hoping everybody will at least be taking written notes either on paper or on the tablet where this blank slide is. Yeah, you won't have this in the PDF file. Now it only exists in the movie that I post to YouTube after class. How many copies of, or how many molecules of DNA have I circled in blue? How many double helices are in there? Okay, this is going to be a good discussion to have. So why are some people saying 4? 4 is the right answer. And then everybody says, oh shit, I thought I understood genetics. So let me add a different picture. Or if it's not X-shaped like this, when you see an object, it's 2 double helices. So there's 2 circled in red. They're just right next to each other. You can't actually visibly distinguish the 2 double helices. And we'll talk about this because this is a really important point that is always unclear, but I want to make sure it's as clear as possible. So that was a human karyogram. Let's look at gorillas, just for fun. Okay. Homologs, homologous chromosomes, circled in blue. There's gorilla chromosome 1. 2 homologs. Chromosome 1 that gorilla inherited from its dad and 1 from its mom. So how many molecules of DNA or how many double helices are circled there? 4. You can see them. Right? If we blew that up, if I made an ideogram, that object on the left that looks like a really elongated X is 2 double helices that touch at the centromere. Right? So there's a centromere right there. That drawing is meant to represent that thing. Right? It's 2 double helices. It's an X-shaped kind of thing. So that's 4 molecules of DNA, 4 double helices. And there I'm circling another 2 double helices. So anytime you see something that looks like we think that's a chromosome, 2 double helices.
So these have an obvious centromere?
Right.
The human one didn't have an obvious centromere?
So that picture we were just looking at, you just can't see where the centromeres are. Right. Yes. So all chromosomes have centromeres. It's just that early in the cell cycle, you can't tell where they are by looking at the chromosome. It's later at this stage you can. Please.
This is
Bless
for
you.
a diploid organism. For a tetraploid organism, then we would have like the 4 copies in each one.
Right, so if this was a tetraploid organism, it would have how many homologs?
4 homologs.
It would have,
I mean, 4, yeah, I don't know.
there would be 4 homologous chromosomes. You'd see something like that
Yeah.
in a karyogram. So at metaphase, a tetraploid, how many double helices is that? 8. 8 D helices. Yep.
Do scientists actually have like the karyogram for a tetraploid?
Yes. Yeah. So you could Google it or do a web search for it. Tetraploid organism, then find out the name of a tetraploid organism, then search for organism name karyogram. See what pops up. My guess is salmon. We talked about salmon briefly, but salmon tend to be polyploid. They have multiple copies of a chromosome. Okay. A couple other points to briefly reinforce. So somebody brought up centromeres. How many different names do we have for centromere positions on chromosomes? How many was that? I heard 4. Who said that? I'm going to make you tell me all 4. No, I'm just kidding. So we name chromosomes based on the location of the centromere along from top to bottom. Okay. What's chromosome 12? Where would you say that its centromere is located? If you just described it to me verbally. It's in the center. It's about halfway from top to bottom. So that's called a something-centric chromosome. Meta, for middle. So that's a metacentric chromosome. Anytime the chromosome is divided about in half, the position of the centromere is right in the middle, it's a metacentric chromosome. Let's see. What about chromosome 7? It's hard to tell. In fact, I don't even know if I really believe that those are homologous chromosomes. Do you? One of the ways we know which chromosomes are homologous visually is the length of the chromosome and its position of its centromere. So do those two look like they have the same length and centricity? Position of the centromere, length, are they the same or not? This is highly debatable. There is no right answer. But this is the sort of question I might ask you on an exam to help test your understanding of how we do chromosome nomenclature. Pick chromosome 7 and tell me, do you think that those are homologous chromosomes or not? If not, why? Go ahead.
If those, so if this is due to cell division, could it have been during replication too if one had a duplication of a gene zone that elongated, but it's still meant to be with chromosome 7?
Fair enough. So let's say, for example, which chromosome 7, let's say just arbitrarily that the one on the left is paternal and the one on the right is maternal. What could happen to the paternal copy to make it look different from the maternal copy?
Could have had an insertion.
Which one's longer, P or M? P. Okay. M is what sort of centricity? It looks pretty metacentric. Centromere is right about in the middle. What about the P? The paternal chromosome, that is. This is what we call submeta. It's slightly off-center, submetacentric. That's a B. So if you wanted to, and hey, this is going to sound familiar. If you wanted to explain how those two chromosomes are different, what could have happened? Could there have been an insertion, was the question. So my question to you then is, how would a duplication of DNA, how would a duplication explain this? Where would it have to happen? Where would DNA content have to be duplicated to make these two homologs, if they really are homologs, look different?
In the bottom, underneath the centromere.
Okay, so what if there's some extra DNA here that's a duplication of something else that was on that arm of the the paternal chromosome? So you copy and paste DNA right next to itself, makes that arm of the chromosome a little bit longer, which also makes the chromosome then look like it's no longer in the middle of the chromosome. Alternatively, you could imagine that maybe the P version is the original version. What if there was a deletion of that material from dad's copy relative to dad's copy? Maybe mom has a deletion of part of this chromosome, and she passed that on to this individual, their offspring. And so maybe, how would we know that these are still both chromosome 7s then? They look different.
Relative position of genes.
So we'd need to know something about the genes. So it might be, for example, that the short arms all contain exactly the same genes. I'm not going to make up gene names. We'll just call them genes A, B, C, and D. So if we knew that genes A, B, C, and D were both on the short arms of those chromosomes, and we knew that genes F and G were still down here on the bottoms of the chromosomes, that would be enough information for us to decide, yes, those are homologous chromosomes. They're the same chromosomes because they have the same genes on them. It's just that they look different in size because there's also been a deletion or an insertion. When you have a submetacentric chromosome, the chromosome arms aren't the same length. This is the last nomenclature-ish thing. We have specific terms for the short and the long arms. What are they? P and Q. Mind your P's and Q's. What is the P arm? Let's talk about chromosome 8 then. Which one's the P arm? P is the short arm. Q is the long arm. And how do you remember this? Supposedly, P stands for petite, small. And I've heard that that may not actually be true, that that's just a mnemonic somebody made up to remember, but whatever, now you've got it too. What I heard after that was that Q stood for the word queue, or long line, like you're queuing up to watch a movie, standing in the line to get tickets or something like that. Regardless, P, short arm, Q, long arm, they only exist for submetacentric chromosomes, where the centromere is slightly off-center. Obviously, there's no longer and shorter arm if it's metacentric, by definition. And then we have two other types of chromosomes, acrocentrics and telocentrics. So acrocentrics, maybe chromosome 18, for example, where there's just a tiny little nub of short arm, barely some DNA you can see above the centromere. It's an acrocentric. Telocentrics basically have no short arm at all. It's telomere in order down the chromosome. Telomere, centromere, coding DNA, that is the stuff, the genes that makes up the DNA, and then, or makes up the chromosome, and then another telomere. So when the telomere and the centromere are right next to each other, those are telocentric. Yeah?
As far as choreograms go, homologous structures are not always paired.
So they are. Well, let me ask you to clarify a bit. You mean, one way I could interpret that question is this the way chromosomes look when you look at a nucleus? Is that what you're asking?
No, but you said that some of the structures on, like, number 7 are not homologous, so on a karyogram they're not, even though they're matched up, they're not all homologous
They're homologous chromosomes. So we've paired chromosomes that have the same DNA content that we inherited from our dads and our moms. But there are some times when you look at an individual's chromosomes, be it a plant, animal, whatever, and they don't quite look exactly the same. What's an example of that? I try to come up with one here where you imagine that one parent has a deletion of part of their chromosome and another one doesn't. So when you have their kid, obviously the chromosomes aren't quite going to look the same, even though they're both copies of chromosome 7. What are other situations where you have chromosomes that don't match when you look at an individual's chromosome? Yeah, the X and the Y chromosome. Presumably I've got a Y chromosome I got from my dad, and that's why I'm male, I think. And an X chromosome that I got from my mom. They definitely don't look the same. They don't even have the same genes, but they're considered homologs because they're the 23rd pair.
What was acrocentric?
Acrocentric is when you can see just a tiny little bit of DNA. You would consider it to be a short arm, but it's barely visible. In practice, for this class, I will not ask you to distinguish acrocentrics and submetacentrics because that's very qualitative. How small does the short arm have to be before you call it an acrocentric chromosome? I don't mind, or I don't care at this point. So metacentric, centromeres in the middle. Telocentric, the centromeres at the very end. Anything in between you can call acrocentric or submetacentric. I don't mind. And there isn't a really good example of a telocentric pair on here, so I'm going to draw one. Here's an ideogram of a telocentric, what it would look like. Right. No short arm at all. So it looks like a V, or an upside down V. And again, each of those lines actually representing a double helix.